I'm John Teem. I'm a Senior Scientific Program Manager for the ILSI Research Foundation. And I'm not a researcher doing synthetic biology. However, I was involved with organizing this session, and in the course of doing that, I became familiar with a number of different stories that are taking place that involve synthetic biology and agricultural biotechnology. So I wanted to share those stories with you this morning and introduce this topic of synthetic biology, which may be transformative in certain areas of biotechnology. So as we go through some of these examples, I'd like you to think about that in terms of how might this change what I'm doing? How might it change the type of research that I do if you're a researcher, or how might it change the regulatory framework that I'm involved with if you're a regulator? Oh. So what exactly is synthetic biology? I pulled this definition off of the internet, and I think it's actually a pretty good definition. Synthetic biology is the design and construction of new biological entities such as enzymes, genetic circuits, and cells, or the redesign of existing biological systems. So that actually covers a very wide range of topics. But I think another aspect of synthetic biology that is different is the perspective of biological machines. So as researchers, we oftentimes are concerned with trying to figure out biological systems and understand them. And in synthetic biology, it's actually a very different perspective. The perspective is one of understanding biological systems, but from the standpoint of an engineer, to try to make use of the various parts of that system to create something that never existed before. So another difference, I think, associated with synthetic biology is the notion of scale. So we're all familiar with the process by which genes are introduced into plants, as an example, and how we are normally introducing a single gene in order to produce a new trait in that plant. So the scale is different for synthetic biology. It may be that we're trying to introduce a number of genes that will be functioning in a coordinated fashion to produce a new trait. So we may be introducing an entire pathway as opposed to a single gene. And in doing so, we're trying to take that pathway and turn it into a set of components that could be used to introduce that same pathway into a variety of different organisms. So in this presentation, I'm going to talk about this notion of interchangeable genetic components or parts. And I'll talk as well about an organization called IGEM, which has been involved with synthetic biology with regard to getting students interested in this topic, and as well producing the types of parts that would be useful in the construction of a biological machine. And then I'll talk about 3 examples where we're interested in trying to introduce a pathway from one organism into another. So the first example will be the introduction of a plant pathway into yeast. And then the second will be introduction of a pathway that occurs in bacteria, but into plants. And then lastly, a pathway from one plant or one type of plant into another type of plant. So we're all familiar with the structure of genes, and genes have characteristic features such as the promoter and the coding sequence and the terminator, which stops transcription. And so in a synthetic biology perspective, a gene is a part. It consists of these different regulatory features that can be characterized so that we know that we'll get a predictable amount of expression from that part, from that gene. And so in synthetic biology, we're basically concerned with producing a toolbox that consists of genes where we have a known output that's expected from that gene, and we're going to then put those component parts together to produce a new biological device of some sort. Now, one way that this has been done, and I'll just talk about this briefly, is to try to make use of the gene as a form of an electronic component resembling a transistor. So a transistor is a device that can exist in two different states. And so as a result of the efforts of Drew Endy at Stanford University, there's been a large effort to try to turn genes into components that could be used to construct logic gates. And ultimately, the goal would be to try to put those logic gates together to construct a microprocessor. And if that were possible, it would be further possible to program cells to do things that they wouldn't normally do. So this is a very interesting aspect of synthetic biology, the notion that we can basically program cells ultimately for certain types of gene expression and get new traits and features as a consequence of that activity. And he actually termed this a transcriptor, which is analogous to a transistor that you would identify as a similar type of component in electronics. So what I'm more interested in talking about today is the use of synthetic biology to string together a series of genes that might be involved in a metabolic pathway, and then constructing a new biological device to have some particular output that you could transfer from one system to another. So in order to do this, from a synthetic biology point of view, what's necessary is to recode the genes so that they are going to be functional units that can be deployed in a variety of different types of host organisms. So recoding means taking away the regulatory features that might normally be associated with those genes in their native environment. And then stringing those genes together with the appropriate promoters and terminators so that all of the enzymes in the pathway are produced and give rise to an output at the end of the pathway that resembles what occurred in the original host organism. Now, this is a complicated process because in removing those regulatory features, we're going to change the flux of material through the pathway. So the promoters and terminators have to be precise in their strength in order to produce the respective enzymes in the pathway in the amounts that are needed in order for the material to proceed through the pathway to the end. So in synthetic biology, there's a real importance associated with getting a predictable gene expression associated with various promoters. And these promoters are considered to be parts in the machine that can be used to precisely regulate a pathway. Now, another complexity associated with this is that if we're moving a pathway from one organism to another, some enzymes in that pathway might require a specific environment within the cell. And so it's important to consider where will that product end up in the cell, what microenvironment. So it might require, for example, an acidic environment or an anaerobic environment. So targeting has to be considered with regard to different parts of the pathway to make sure that the enzymes end up in the correct microenvironment. So what I'd like to consider first, before we consider a metabolic pathway, is a signal transduction pathway. And the reason for this is I'm going to later talk about a signal transduction pathway in plants that we'd like to move from one type of plant into another. So this is sort of a template to think about that problem. and what would it take to change a signal transduction pathway to solve a particular problem. So what I've got shown on the slide here is a signal transduction pathway that takes place in bacteria. So it's the chemotaxis signal transduction pathway by which bacteria sense attractants in the environment, and then a signal is sent when that attractant binds to a membrane protein on the outer membrane of the bacteria, and the signal is sent ultimately to the flagella, which controls the way the timing of the flagella in its operation. So an attractant then serves to activate the signal transduction pathway, and the output at the end is a change in the motion of the flagella. So this is a basic template that can be modified using synthetic biology to change the attractant and also to change the output at the end of the signal transduction pathway. So the attractant can be changed to a whole variety of different molecules. So as an example, arsenic. The binding of arsenic can be used to activate the signal transduction pathway by making a hybrid membrane protein that'll bind arsenic and send the signal. Or estrogen, another small molecule that you would never find as part of a normal signal transduction pathway in bacteria, but we can modify this existing pathway to respond to that human hormone. And then we can change the output as well at the other end of the pathway. So instead of having a change in flagellar motion, we can have a reporter gene, which activates green fluorescent protein, something of that sort. So the basic notion here is that we can make biosensors by manipulating this signal transduction pathway. And we have to change it at both ends. We have to change it at the point where the signal molecule binds to the receptor, and also at the output portion. Now, one of the reasons why I use this as an example is that this type of biosensor machine has been generated many, 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 many times. And it's been generated through the activities of this group, IGEM. So the IGEM competition is a competition that involves synthetic biology and tries to bring students into this process to try to create different types of biological machines, like a biosensor, as an example. So the way this organization works is that students will come up with an idea to try to make a synthetic biology machine. They will register with the IGEM community, and then they'll be sent a distribution package which contains in it the different parts that they would need to construct their machine. So in association with a laboratory, they'll make their machine, they'll get their results, and then they'll meet at a jamboree, which is a large meeting that brings all the teams together to compare their results, and there'll be a winner, and they'll then turn their parts into the registry where they initially obtain their distribution kit. So the importance of this is that every year that this competition takes place, there are new machines that are made and new parts, and the new parts are added to the registry so that there are even more parts available for the next year's competition. So in 2006, there were 167 parts. And now in 2017, there's over 20,000 parts. And this has been a very productive approach to try to generate the means to fill the toolbox with parts. Now, I want to then at this point, go through some different examples involving synthetic biology. And the next one that I'm going to go through is not a result of the IGEM competition, but rather the results of a laboratory that's focused on synthetic biology for the purpose of doing an alternative path for the production of opiate drugs. So in this particular example, this is work from Christina Smolke at Stanford University. What she's done is she's taken a metabolic pathway that occurs in poppies, and then taken all the genes involved in that pathway, appropriately constructed them into parts that can be assembled into yeast to get the production of an opiate at the end of that pathway. So in this case, the opiate is an intermediate thebaine, which is required for the synthesis of morphine. So in this example, we're actually going from the direction of plants to microorganisms, trying to take a pathway that would normally exist in a plant and then move it to the yeast, Saccharomyces cerevisiae. And in this case, it was successful. It was possible to generate the opiate, but in very, very small amounts. And part of the reason for that is that there are a number of problems that exist in moving a pathway of this sort that have to do with the misfolding of proteins that require a specific type of glycosylation, or getting the proper amount of synthesis of each of the intermediates in the pathway. So these are all solvable problems. So as engineers, it'll be possible to increase the output from this pathway. But I just wanted to introduce it as one example of a system where the pathway normally exists in plants and now it's being moved to another organism. I want to now consider a very different type of pathway which is relevant to agricultural biotechnology, and that is nitrogen fixation. So the problem that is being addressed here is the problem that plants require fixed nitrogen in order to increase the agricultural productivity. And so that requires fertilizer to be applied to the fields, and fertilizer is expensive, and it also causes all kinds of problems if it runs off into the surface water and can lead to algal blooms and a number of other problems. So there's a need for fixed nitrogen that is a requirement for productivity in agriculture. And we're currently supplying that need with fertilizer. But it would be very useful if plants could provide their own fixed nitrogen. And the way that plants get around this problem is that they form an association with symbiotic bacteria which do the nitrogen fixation for them. And so certain types of plants are capable of this type of symbiosis, and they will respond to signals that they get from bacteria in the environment to form root nodules. And in the root nodules, the bacteria can infect the structure and become associated with an environment where nitrogen fixation can take place. So this is a process that normally takes place in the bacteria, but a synthetic biology solution to this problem would be to take those genes from the bacteria and express them directly in the plant. So the plant is doing the nitrogen fixation itself. So how would that work, and is that possible? So what I've shown here is a slide where a number of genes that are involved in the production of nitrogenase, one of these enzymes that's involved in nitrogen fixation. There are a multitude of genes that are required to produce nitrogenase. And this group here, this is a slide from Luis Rubio in Spain, where they've undertaken this quest to try to express these genes ultimately in plants. But before it can be done in plants, they're making use of the yeast Saccharomyces cerevisiae as a sort of intermediate to allow them to explore the possibility of doing this assembly of this complex enzyme in a heterologous host system. So they've constructed a yeast strain that has various NIF genes that have been recoded so that they're produced in yeast and targeted and specific to the mitochondria. So the reason for that is that nitrogenase is very sensitive to oxygen. So it was necessary to target the components of nitrogenase to an organelle within the yeast where oxygen levels would be low. So since mitochondria are using oxygen at a high rate to produce ATP, that was a suitable environment. So after having introduced these genes into yeast and getting them to express properly, these researchers were able to demonstrate that they could get proper association of the subunits to form tetramers, which is required for activity. But unfortunately, the purified enzyme that they obtained from the mitochondria did not have nitrogenase activity. So there's still something missing here. And so there'll be additional work to try to identify what's the missing component. It might be another NIF gene that's required for assembly, possibly a chaperone that's required. So this is one strategy to try to introduce nitrogen fixation into plants, trying to clone the nitrogen fixation genes, put them into plants directly. Another strategy involves synthetic biology, but from a very different perspective. And that is to try to focus on the fact that some plants have this ability to form this symbiosis with rhizobia, the bacteria that do the nitrogen fixation. Other plants are unable to make that association. So legumes are able to associate with rhizobia, but cereals are not. So as an alternative mechanism to try to solve this problem using synthetic biology, a new approach is to try to make cereals able to respond to rhizobia and form this symbiotic association and therefore acquire that benefit from the bacteria, which they wouldn't normally be able to do. So this again involves identifying genes and then stringing them together and introducing them into cereals, and the genes would be coming from legumes in order to try to transfer this signal transduction pathway from legumes into cereals. So this is a complicated process, and there's likely to be a large number of genes involved. So there are 4 important steps in this process. One is the recognition of the nod factors that the bacteria produce. So there's a small molecule that's produced by the rhizobia. The plant is able to respond to that signal, and it initiates a signal transduction cascade that results in gene expression. And as a result of that gene expression, there is the formation of the nodule, the infection of the bacteria into the nodule, and then the production of the suitable environment for nitrogen fixation. So there are a number of genes that are involved in all steps of this pathway. So the challenge here is to try to reconstruct this entire pathway that occurs in legumes in cereals. So how can that be done? Well, fortunately, one aspect of this pathway is already in existence in cereals. So there is a pathway called the SIM pathway, which already exists in cereals, and it's the same pathway that occurs in nodule formulation or formation in legumes. Okay, so they have this common element between them. So that portion of the pathway doesn't have to be reconstructed in cereals, but the front end and the back end do have to be reconstructed in cereals. So it's going to be necessary to make cereals respond to the nod factor and then as an output start to do gene expression to produce the appropriate gene products required for nodule formation. So the basic idea here is that we're retaining the use of the SIM pathway, the signal transduction pathway, but changing the response of that pathway to be specific for nodule formulation factors. Now, I should mention that the cereals have this pathway because they respond to a different set of signals, signals that are produced by fungi for mycorrhizal symbiosis. So the idea here is that we're substituting signal molecules and then ultimately changing the final output of the pathway to allow nodule formulation instead of genes associated with mycorrhizal expression. So again, this involves putting together a whole string of genes that would be involved at the front end of the pathway to respond to the nodule factors, and then at the other end of the pathway to to respond to the signal transduction pathway for activation of nodule gene expression components. Now, some of the genes that are involved at both ends of this pathway are known, and I've listed some of these on the slide here. It's a very similar type of situation as what I described in the beginning with the bacterial signal transduction system. There is a receptor. It's known to exist in legumes. It's presumed to exist in cereals. So there'll be additional research required to identify what those corresponding receptors are. There'll be additional work to identify what are the genes involved in nodule formulation in legumes to engineer those into cereals. So just as in the first example where I was talking about a solution to this problem involving nitrogen fixation genes, in this particular example where we're trying to manipulate signal transduction, we're also at an intermediate stage. It hasn't yet been taken to the point where it seems feasible that cereals will ultimately be engineered into nitrogen fixing plants. So more work is needed in both of these strategies, both of these synthetic biology strategies. And additionally, there'll be additional work that's needed to understand how surrogate systems like yeast can be employed to transfer these pathways first to yeast to do the troubleshooting and then ultimately to plants. And just as a general feature of synthetic biology as it applies to agricultural biotechnology, there's going to be a need to fill that toolbox in some fashion. So I mentioned how the IGEM project was very successful in generating tools for building biological machines in bacteria. So a similar type of effort is needed to assemble a toolbox for plant molecular biologists to put together these types of synthetic biology projects for the benefit of agriculture. Now, the last thing I want to consider here has to do with risk analysis for synthetic biology organisms. And the question that comes up is, do we have to do something different here with respect to organisms that employ synthetic biology as compared to the types of traditional events that we're considering now using existing technology? And I would argue that we don't need a new risk analysis framework to consider these organisms, because the same types of genes that are used to make conventional genetic crops, genetically modified crops, there are the same genes that are being used for synthetic biology. So those parts haven't changed. And we're able to do risk analysis and risk assessment with the tools that we have in hand for both genetically modified crops as they exist now, and as well for synthetic biology crops that are produced in the future. Thank you.